Hi students, we had earlier spoken about immunoprophylaxis and the history of immunoprophylaxis, and we came across the term waxen always. We told that vaccines have been developed for the immunoprophylaxis. But I want to ask you something. Do you understand basically what is a vaccine? A vaccine is a medical preparation which has been given to provide immunity from a disease. They use a variety of substances ranging from dead microbes to genetically engineered antigens to defend the body against potentially harmful microbes. And the effective vaccines, they can change the immune system by promoting the development of antibodies that can quickly and effectively attack the disease causing microbes when it enters the body, and thereby preventing the disease development. Now the vaccine, they can contain live attenuated or even killed microbes or parts or products of the microbes such a way that they are capable of stimulating a specific immune response comprised of protective antibodies as a T cell immunity. So any preparation of a bacteria or a part of it or a genetically engineered part of it which when inoculated into our body can induce an immune response and help us to protect against or defend against the disease development is referred to as a vaccine. Now, another thing that we have to notice about a vaccine is an effective vaccine is something which would trigger an immune response in us. What is an immune response? An immune response is nothing but the production of active B lymphocytes or the effector B lymphocytes as well as the effector T cells. Okay, so these are being formed mainly from the memory B cells as well as the memory T cells. So if a vaccine, what is a vaccine? A product, a part of the microbe which is either live or inactivated, or which is a genetically engineered product, or whatever it is, or it can also be a toxin which has been inactivated. Okay, if such a vaccine can trigger an immune response by waking up the memory B cells and the T lymphocytes to become active antibody producer cells as well as effector T cells, then such a combination is referred to as a vaccine. Now, if it is a viral vaccine, we expect to get high titers of neutralizing antibodies when we give a viral vaccine. And usually when the injection of a vaccine has been done into a non-immune subject, what happens? It should provide an active immunity against the modified pathogens. And vaccination thus is a immunization against infectious diseases through the administration of vaccines for the production of active immunity in humans or other animals. Now, what are vaccines? There are different types of vaccines that we have. Now, the first type we are talking about is a live attenuated vaccine. The live attenuated vaccine is a vaccine which is being it is of course live, but it has a reduced virulence, okay, and but it also has a immunogenicity. And this live attenuated vaccines, okay, they are basically this attenuation is a process which can be achieved by different methods, like you can either grow them in hostile conditions so that the bacteria or the pathogen, whatever it is, that will be able to grow rapidly. Okay, it can grow probably, but it cannot cause an infection. Okay, it will be like you are making the person tying the bacteria and putting it, like it can, okay, you can enter, you can go inside, you can be survive over there in the host, but it cannot cause an infection. That is a case of live attenuation. About how we are attaining live attenuation we will be talking in detail, but I am just giving you an inclusion about what are the different vaccines that we have. Now here, live attenuated vaccines, we have tuberculosis, for example BCG is a vaccine, oral polio vaccine, measles vaccine, the rotavirus vaccine, as well as the yellow fever vaccine. These all are mainly the live attenuated vaccines. Coming to the next one we have is the inactivated or the killed antigen. Here what happens? The total microbe is being taken and you kill it. You kill it in such a way that it cannot multiply. Okay, it is like a dead cell which has been given inside you, but because of the presence of certain molecules on these killed molecules, certain epitopes, okay, certain epitopes of the antigen will be identified by our own host immune response, and that would host cell, and that will trigger an immune response within us. Such inactivated killed vaccines are included in whole cell pertussis as well as inactivated polio vaccine. Coming to the next one that we have is the subunit or the purified antigen. Now in this, what does it mean? You take some small subunits, as the term implies itself, small parts of the microbe or the virus or bacteria or the virus which we have. You take small part of that, and they are being purified, and they are being given as the as a immunogen as a vaccine. They mainly are being used now. For example, the vaccines for acellular pertussis AP, the Haemophilus influenza type B Hib vaccine, the pneumococcal vaccine, as well as the hepatitis B vaccine. These are some examples of that. Then another thing that we have is a toxoid or the inactivated toxin. Okay, this is a toxin of the virus which has been of the bacteria or the virus. And now most probably it is the bacteria which is in the case of a toxoid, because most of the toxin producing ones are bacteria. Now here we have the tetanus toxin as well as the diphtheria toxin. These have been taken and they have been inactivated, and they are injected within us. You might know the vaccine of TT, how it is. When we are injecting it on our hands, what happens? You will get a highly severe pain over there, and unless and until that is an inactivated toxin that which we are injecting, and unless and until we spread it or massage our hands completely, that that toxoid is not being transferred throughout. You move your hands rapidly, the toxoid will be spread over, and the effect of it would be found to be reduced. Then another things, now look at the picture, you can find you have recombinant vaccines. Okay, recombinant vaccines, these are nothing but now the first example that they have shown here, see, just look at this one, a recombinant subunit viral protein. That is, some part of the protein of the virus is being put into a recombinant vector. A vector is, so the gene which has been coding for the particular protein, virus protein, is put into a vector. You know what's a vector? A vector is something which can carry a part of DNA and which can rapidly, so you produce a recombinant, a part of, now for example a plasmid is a vector. You take a plasmid and you take the gene for the virus coding protein, and you produce a recombinant plasmid, and then further you multiply it, and then you will produce what happens, such proteins, the recombinant viral proteins might be taken. And another one, same way, that's a recombinant subunit viral protein. Okay, protein, and that protein might be further mixed with certain other subunits, or it might not be one single part, it could be different subunits of the virus which is being used as, so that's what that is also been possible. Okay, so you take a single subunit or a multiple subunit depending upon the immunogenistic property. So you take a small part of the virus, put it into a vector, multiply it in a sense, let it grow for several stages, and then further you extract the particular protein as well as, and you use it as a vaccine. That is a recombinant subunit viral protein. Now the same way you can also use it for the case of bacteria, recombinant bacterial vector. Okay, here what is it? It is a bacterial vaccine vector which is being used here. Then next one, you have virus-like particles. They are not virus, but they are virus-like particles which can be used as vaccines. Then you have the DNA, small segments of DNA. They would carry the viral genes in them. Okay, and so that the DNA as such which carries the viral genes can be used as vaccines. They are the DNA vaccines. Then you have recombinant viral vector. Okay, the vector that you use here is a virus, not a bacteria. Okay, in this case here you have the recombinant bacterial vector, that is, the carrying agent was a bacteria. Okay, so one particular protein of this virus was taken, and that gene was put into a vector which is a bacterial vector in this case, and here a part of the protein of the virus has been taken and it is put into a vector which is a virus, and the virus will multiply and produce large amount of course this virus will be something which will not be toxic for us. Okay, then you have synthetic peptides. What does it mean? If you have a particular protein on this virus, okay, I will take that protein, I will sequence that protein, and I will know the amino acid sequences of that. And what I will do by synthetic chemistry, I can synthesize that particular peptide. It would be a small subunit of the, not the whole antigen as we consider, might be one particular protein of that antigen would be immunogenic. So that's what you take, and you might take a synthetic peptide, and that synthetic peptide could be injected within our body. Of course there would be some formulations, of course, because I have mentioned about earlier in the previous classes, we had mentioned about adjuvants which would be probably increase the immunogenicity of a vaccine and all. So all these things probably they are being created based upon different principles, but their main purpose is to increase the immunogenicity or to able to trigger your immune response in your body without causing an infection in you. So these are the different types of vaccines which I can explain to you, which I've explained in a brief manner. Hope so you are clear with it, and thank you. The details of individual type of vaccines we will be talking later. Thank you for the session.